speaking of things that are crazy, I don't know if anyone else feels like this, but it's not a problem, but maybe it is. The fact that we have just little tiny machines inside of us that do everything and keep us alive it sounds like a crazy statement, but that's literally just what's going on. It's just normal. It's just what's going on in our bodies. And their motorcycle. There's little motorcycles. Oh my god. They are some of them are motorcycles though. Oh, I hate that. Oh, I hate that so much. God damn it. There's little motorcycles inside of us that do everything, and everything that I've ever talked about in all of my lectures is, they all involve the underlying motorcycles inside of us. No matter what it's been, neuroscience, pathology, drugs, endocrinology, everything is motorcycles. Everything is machines, everything is proteins. Um, and each time, no matter what it is, I always demonstrate how molecular biology is lurking beneath all of it. Um, I, I showed you how insulin, a protein, affects the different enzymes, also proteins, that break down sugar. I showed you how vitamin D is made through enzymes. We talked about the different receptors, proteins, that help serotonin carry out its function. Prions are proteins that are infectious, and they arise from a protein that we already make normally. We even talked about Sonic Hedgehog, which is a protein that directs our limb development. And finally, I did a lecture on epigenetics, which is just really probably the most on the nose molecular biology lecture that I have done. But we're gonna we're gonna delete it. Well, we're not gonna delete everything, but we're gonna push all of those things away, and we're gonna go back to the foundation of molecular biology, and we're gonna look at how life itself is orchestrated. Like the reason motorcycles. Matt, would you ever name a child motorcycle? Oh wait, no, you're not Matt. Oh my god, it's Austin. Austin definitely would name a kid motorcycle. That's what we were talking about earlier. Is I would I would name I would name my child Karyotype, and then Amy would name our other child motorcycle. Yeah, she just would. Uh, and then we learned that there are motorcycles inside of us right now that are directing our existence. Just literal motorcycles that are machines that are the reason why we breathe and sweat and talk and speak and digest and shit. All of the things that our body can possibly do are because of proteins. Um, Motorcycles. And molecular biology, uh yes, the motorcycles inside us. Oh my god. Now, because I always talk about the machines inside of us when I'm, talking about proteins, but now I can't ever it's motorcycles every time now. Holy shit. Motorcycle biology, that's what it is. Uh, but, molecular bi biology in general, like, it's broken up, right? There's a lot of different disciplines within biology, and molecular biology itself is also prone to those, divisions. So right here, this is sort of the relationship between molecular biology and some other related disciplines, which are, in essence, also molecular biology. Don't at me, geneticists or biochemists. You're just molecular biology. But these guys really shake hands with each other because our form and function is determined by information that is stored within our genes. And then those genes, the information within the genes is used to make proteins. And then, of course, the proteins are what designate our form and function. It's all one big circle, all one big motor cycle, if you will. And this is all life in the whole entire universe, not just us. It's every single type of life, from humans to hydrangeas, mutts, mushrooms, fucking crabs, chlamydia. Every every single life fucking does this. Like, goes through this. And this very idea that genes are making proteins that carry out all of our life's processes, this is called the central dogma of molecular biology. Uh, this is everything. This is who we are, what we are. This is what our cells are doing 24/7, taking our genetic blueprints and making everything that they need and everything that we need. And like, that can look like anything. It could be an enzyme to help us metabolize things, a hormone to help us our cells communicate, or receptors in order to facilitate communication. Every possible thing. Um, and I mean, these 5 words and 2 arrows, it's everything. I'm not gonna cry. uh, every time our cells need to make a protein for whatever reason, metabolism, gene regulation, what they're gonna do is they're gonna access our DNA, our life blueprints. They're gonna turn it into RNA, which is like a middleman, and then that RNA is gonna be made into a protein. Well, this is Miss Frizzle. This this is high school biology, right? We all know this. This is not a new concept, but we're not stopping here because we're all going on the magic school bus, and I'm gonna Miss Frizzle you. We're gonna go as small as we possibly can, and we're gonna ride the little tiny motorcycles inside of us. We're gonna do it. That's what we're gonna do. So get excited. Like, get just get fucking pumped. Um, so let's just get right into it. Step 1, transcription. We're gonna transcribe our DNA into RNA. So let's take a look at the structures of these beautiful chemicals, and then we're gonna explore the process of DNA being transcribed into RNA. Um, I mean, that's just me. That's me. Um, this is familiar. We know what this is. DNA. Uh, it's a double helix. It's double stranded because our genetic code is vitally important. So, in order to keep it really safe, we like to have it in 2 complementary forms. Um, Classic. This is also DNA. Chromosomes. A single chromosome is just one really fucking long string of DNA that's been really, 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 really wound up super, super, super tight. So chromosomes are DNA. If we go a little further, we're gonna do a lot of this tonight. We're gonna do a lot of this is a unit of this, and this is a unit of this, because that's just fucking biology. But an individual unit, basically, of a chromosome is chromatin. Chromosomes are made of chromatin. And chromatin just refers to the combination of DNA and proteins all together. So this right here, this is a chromatin fiber right here. And this string, of course, is DNA. And it's being wrapped around these spools, called histones. Histones are proteins. It's a group of proteins, and that histones are what DNA physically wraps around. And right here, this is a pretty closed conformation. This is pretty tightly wound. Um, A unit of chromatin is nucleosomes. A nucleosome is just this one part right here. I think it's specifically like 146 nucleotides of DNA wrapped around a histone core. It's wrapped about 2 times. And right off the bat, I would just want to tell you, this structure right here is very tight. So the genes in there are going to be pretty inaccessible. But here, where we can really see the beads on a string a little better, and they're farther apart, these genes are going to be a little more accessible. And this this is a really good representation of what I talked about, but I want to go over the vocab really quick anyway because it's kind of fucked up. So, because all these are just DNA. These are just fancy ways of different ways of saying DNA. A chromosome is one big fucking long DNA that's been tightly wound. So that our there's like 6 feet of DNA inside of each of our cells. So putting it into a chromosome is a way to ensure that that whole fucking 6 feet of DNA can adequately fit inside of just the nucleus of our cell. And then chromatin is just a unit of a chromosome, the actual complex of DNA and proteins together. We have heterochromatin, which is really tight, close together. Not a lot of transcription going on, not a lot of genetic access there. And then we have euchromatin, farther apart between the nucleosomes, much more open, more transcription, more proteins being made from those genes. And then the nucleosome, I have 2 representations here. This is just showing like, I don't know, it's a more canonical depiction of the DNA wrapping around the proteins. The proteins being histones. Just 8 slices of pie, basically, together to form the spool that DNA wraps around. And this is a big thing that I talked about in the epigenetics lecture, is that you can make changes to the histone in order to open or close the genes for transcription. So that's just what I have here. Usually if you methylate them, you close them up, and if you acetylate them, you open them up. I'm not going to go too much into detail about that, but it is important to know that like this is kind of going to be the theme of the lecture of proteins can interact with each other in a lot of different ways. And this can affect basically the course of the central dogma, as we're going to see. So, yay vocab. Oh yeah, prokaryotes, like bacteria, they do not do this shit at all. Only eukaryotes do that. And that's us. We are multicellular eukaryotes. So this manner of packing our DNA into chromosomes and like having all this epigenetic modification, this is an us thing. I'm not really going to be going into what bacteria do because I don't give a shit about that. No. I just think that this is a lot more like fascinating. And I wanted you to understand that the organization of our genome in this manner, like I said, it affects the dogma. It affects whether DNA is going to proceed to RNA and to protein. So believe it or not, this is zoomed out. We're zoomed out right now. I'm always doing this, right? I'm always doing this. I'm always accessing the genes in my euchromatin. Classic us. This is unbelievably. You thought it was No, Monkey Monday is the first Monday of each month, and then Molecular Monday we do the last Monday of each month. It's a classic mix-up. It's okay. I could talk about monkeys too. You know, monkeys do this. Just like us. But yeah, this is zoomed out unbelievably. We're going to zoom in even more because we need to look at the chemistry. I know we're allergic to that word, but we're gonna look at the chemistry really quick of DNA and RNA. Super quick. So this right here, this is a nucleotide. This is a unit of DNA. Um, uh, my brain. A nucleotide is a unit of DNA. In total, we have 6 billion nucleotides in our genetic. 6 billion. I'm not mad. 6 billion nucleotides. That's a lot. When we measure our DNA, we measure it in base pairs. And that is referring to the fact that we have nitrogenous bases in DNA that each pair with each other because it's double-stranded. A always pairs with T, C always pairs with G in DNA. So that's what the base pairs are referring to. And ATGC, that's the code right there. That's it. That's the code. It's 4 letters. Oh my god, it's 4 letters. I'm not having an existential crisis yet. I can't have it. Gattaca, exactly. Gattaca. Unironically, that is Izzy's favorite movie. And she defends herself by saying Ethan Hawke. Who's the other guy? And Jude Law in the same movie, and I'm like, okay. And Uma Thurman. I like it. It's not bad. And yes, perfect representation of the genetic code right there. I bet I could do a search for all the Gattacas in my code. That'd be really fun. But yes, the code ATGC Gattaca, that's it. That's all it takes is those fucking letters. That's all that's in our code. Now, if we look at this nucleotide, I can tell right out the gate that it is from DNA and not RNA because it does not have an extra OH or hydroxyl group on the sugar. The pentagon right there, that's the ribose sugar. But here, D, as in deoxyribonucleic acid, D means without. So we just don't have one here. That's how I know for a fact that this is DNA and not RNA. We just have our single hydroxyl on the 3' carbon. We also have a phosphate on the 5' carbon. And then this big chunky guy right here is the nitrogenous base. In this case, it's adenine. So this is everything that makes the single nucleotide of DNA. One of the is the building blocks. So when they're together though, like right here, the 2 strands of DNA are joined by hydrogen bonds, which is fantastic because those are pretty easy to break. And we are constantly breaking these bonds so that we can either replicate our DNA or transcribe it. So the hydrogen bonds are for the connect the 2 strands, and then the phosphodiester bonds are what connects like the backbone of the DNA itself. So right here we have this is the 5' phosphate connecting to the 3' OH of another strand. So that is how this is how everything connects. Now DNA, why do we even have this middleman in the first place? Well, that's because DNA is super precious. We want it to stay in the nucleus because the cytoplasm will kill it on site. And we don't want that, obviously. Our genetic code is very important, so we just keep it sequestered in the nucleus forever and ever and ever. RNA can handle the cytoplasm. It's built a little different. So it's allowed to leave the nucleus, and it's allowed to go into the cytoplasm, which is that's where protein synthesis actually happens. So some other differences, obviously DNA is double-stranded, RNA is single-stranded. In DNA, A pairs with thymine, and in RNA it pairs with U. And we think that's probably evolutionary, because this whole early life we think was all RNA. And then we evolved to have DNA. So U is basically a less stable form of T, and that's because of this methyl group right here. This is what makes thymine a little more stable. And also, RNA has the hydroxyl group here. And oxygen is pretty reactive, so this is another thing that makes RNA less stable. So we evolved to have no hydroxyl group there. So now this nucleotide on the left is a lot more stable than the one on the right, which again is important because we have a lot of motivation to keep our DNA protected. So that's why we make an RNA. We make RNA go do our bidding, basically. Now, when it comes time to transcribe our DNA into an RNA, a bunch of proteins are going to converge on the gene that we want to transcribe, and they're going to lead to the creation of a protein. So, proteins make proteins, which means that there are proteins that make proteins that make proteins. I don't hate it. I love it. I sound like I hate it, but I'm not upset at that at all. That doesn't make me think about the little tiny motorcycles inside me too much. It doesn't. I'm fine. All right, so let's do it. We're going to do transcription right now. Loosely, this is a gene. This is a section of DNA. This is the gene that we want to transcribe. So in order to start, the first thing that's going to happen is we're going to have a protein bind to the promoter, and that protein is TF2D. The promoter is the very beginning of a gene. It signals where everything is going to start in order for transcription to take place, and it's very identifiable. I don't think this is always the case, but for the most part, the promoter is just a bunch of T's and A's. Hi, Amy. And we call this the Tata box. That's what we call it. So the very first thing is TF2D binding this Tata box as a way to signal, hey, this promoter is active. This is the gene that we got to transcribe right now, guys. And it's also going to attract and position other proteins to the transcription initiation complex. All right, we need to find a new name for it then. We can't call it the Tata box. Oh, why didn't we just call it the ATAT box? We could just make a Star Wars reference. Molecular biologists love that. All right, before I add to this complex, Amy is here. Amy? Hi. Can I name our daughter Carrie, but it's short for karyotype? No. Like she said no, everyone. Would you name your kid motorcycle? Yes. So no karyotype, which we could hide behind Carrie.
We call her Mo.
Mo? Okay. Like Mo Collins.
Like Monique.
Monique. Monique Recycle. This is why I don't talk to her. God.
Why we separate beds.
Just like Lucy and Ricky. Alright, don't worry, you guys. Amy can't hurt you from here.
I'll be in the chat soon, though. Don't worry.
Oh my god. So, TF2D binds the ATAT box, the promoter, the beginning of the gene. FYI, TF stands for transcription factor or trans factor, and the 2 refers to the fact that RNA polymerase 2 is this protein's favorite enzyme. So a lot of the proteins binding are they're like basically adding to the chain reaction, but some of them do have more specific jobs, like TF2B. This guy is going to come in, interact with TF2D, and help to physically position the RNA polymerase 2 at the start site. Polymerase is what's gonna physically go down the DNA and add nucleotides to the RNA. So very important. We love this guy. We also have TF2F, which again is gonna just help stabilize the polymerase more. We love stabilizing our polymerases. And it's gonna attract more factors. It's gonna contribute to the chain reaction. Lastly, we have TF2H. The H stands for helicase. So along with the polymerase, it's gonna unwind the DNA so that the polymerase can actually move down and physically transcribe the DNA. And it's also gonna help release the polymerase from this complex of proteins, so it's not stuck here. Um, so this is transcription initiation. We're getting everything. We're prepping the poenium, so that we can go into elongation. But there are basically everything is poised to allow the polymerase to do all this, and the reason why that's important is because earlier we saw with the chromatin and the heterochromatin and how sometimes our DNA can be really tightly packed. It takes this chain reaction in order to really allow access to a gene. It takes a village. Um and it takes so many proteins, in fact, that sometimes there are sequences of DNA that are really, really far away from the gene of interest that somehow affect the transcription of the gene that's super far away. And for a long time, scientists were like, what the fuck? How is this happening? Like, it was really confusing. But after a while, we learned that uh, the shape of our DNA really gets changed and affected as these proteins bind. So let's see what that looks like. Again, we have this rectangle is just a strand of DNA that we're trying to transcribe. We have the polymerase bound to all of these factors that we just talked about. And then upstream or behind the gene, far away, we have these other regulatory sequences with transcription factors bound to these as well. And even though they're really far away, they're so the idea is that they're gonna increase the transcription of this gene. And again, this is what was so confusing to scientists until we figured out DNA looping was a thing. Oh, right. I'm supposed to tell you first that these sequences that enhance or increase transcription are called enhancers. And then the proteins that bind to enhancers are called activators. So when these guys all bind, DNA looping is going to occur. So this is that same strand of DNA, but it's all wiggly now. It's the same factors that are bound, but now they're all a lot closer together. So now they can really work in tandem. And we got this big chunky guy called mediator who comes in and sort of clamps everything in place so that all these things can continue interacting with each other and increasing transcription for a longer period of time. It's crazy. This is so this is really cool. Uh it's fascinating to me. That mm, this is basically how if we have proteins where our body just needs a bajillion of them all the time, this is probably what's happening. But the opposite can also be true. Oh wait, I'm ahead of myself. First, we're looking at how this is an example of gene regulation right here, but we're gonna go a little more into it. Yeah, basically, and in order for that gear to even come and help everyone be friends, it's like, oh my god, I'm coming up with a metaphor right now. It's like if me and you and our friend in Europe somewhere or our friend in Australia, if we picked up the phone and called each other at the same time, if that created this thing where we all whoosh, like we're all attracted to one another and like came together as a result of all of us just calling each other, that's what's happening here. Like that's ah, it's it's Neon Genesis. It is. Oh my god, Akira. Holy shit. Yeah. I would definitely watch an anime that was about just transcription. Like, I don't even need other proteins, just transcription factors because they do some fucked up and weird shit. Uh, I think the Cells at Work was pretty close. Cells at Work was pretty good shit, but if whoever's out there, Sakurai or whoever makes Sakurai doesn't make anime. Please make one about transcription factors and please don't call it Tata. Everyone trying to get in these genes. Fuck yeah. Also, ooh, if there were a shorter version of that, it could be the title of the transcription factor anime. Something about getting into genes. But yes, that is essentially what's happening. If you have friends across the world and you all called each other at the same time and that just sucked you all together, that is what's fucking happening here. It's fucking crazy. Oh my god. You just heard sucking.
No, what if we all wanted to like out, but we're all hooking up. We're just like, boom, hangout time.
It would be cool, but also, you won't believe this, but it's happening inside of you right now with all of your motorcycles. Oh my god. Oh my god. We're gonna visualize this in another way. So this is a bunch of nucleosomes, pretty tightly packed together. This is a pretty close formation. The Tata, the promoter, is pretty dug in there, so it's unlikely that transcription is gonna be able to just right out the gate transcribe that gene. I died and reincarnated as a gene transcription regulator. Can't stop creating protein. Oh my god. Yeah, transcription factor Reddit posts. I need that. I need a lot of that. Oh my god. But this closed conformation, we don't need to worry. That's not gonna be closed for long. We got a transcription regulator, and it's binding to something that is related to a sequence that is related to the gene, but it's very far away. That's the anime. Oh my god, the mouthful anime title. All right, I love it. I love it. Oh my god. Yeah, yeah. Isekai. Oh my god. Yeah, in Japanese it's like 2 or 3 syllables. Oh my god. Alright, so our main character, our transcription regulator, um, we'll call him Sakamoto. Uh, because that's my favorite anime, Sakamoto. Sakamoto's gonna open us up, and there's a couple different ways that this can happen. The first is gonna be a chromatin remodeling complex. It's a complex of proteins that it it's in the name. It literally remodels the chromatin, and causes nucleosome sliding. So that spool that the DNA is wrapped around, it just gets slid in a different direction such that the gene is now easier to access. Another thing that can happen is we can recruit histone chaperones. Right here, we just straight up took out the fucking nucleosomes. We just took out the histone proteins, and now we just have this very open string of DNA. Or if we don't want to remove them completely, we could just change the histones a little bit. Remember earlier I showed you it's just 8 slices of pie. That's a histone. It's just 8 slices of pie. If we switch the slices of pie for some different ones, that can change the chemistry such that the chromatin opens up a little more and again exposes the gene so that we can transcribe it easier. And then lastly, and this is very epigenetic, we can recruit histone modifying enzymes. And when we do that, that is also gonna change the conformation because, remember earlier, so histone modifying enzymes, they can add maybe methyl groups to these areas to close them up, or they can add acetyl groups to open them. So also the tags that we put on the outside of the histones are gonna affect how open or closed our chromatin is. Um, so these are just the many, different character developments that our transcription regulator can go through. Um, yeah, so and these are all mainly ways of opening chromatin. But we can't do the opposite. Instead of enhancers, we also have silencers, which are, sequences of DNA that decrease transcription, and then the proteins that bind the silencers are called repressors because they are repressing, transcription. But we don't wanna do this. We're not doing this. We wanna make a protein, so we are just gonna keep, this is what we have right now. We have all of our transcription factors bound, we have our polymerase, we're freaking ready. We're ready to actually turn our DNA into RNA. Um, but there's 2 strands of DNA. So how the hell does this complex of proteins know which strand, to bind to? Because we're not transcribing both strands. We're just doing one of them. And, the answer lies within the trigger warning, chemistry, in the chemistry of DNA. Um, I drew these and they're beautiful and you're gonna appreciate them. This is 3 base pairs of a DNA strand. Um, and I'm showing you that this one starts with a phosphate on the 5' carbon, and this one ends with a hydroxyl on the 3' carbon. I love Khan Academy. Oh my god. I looked up, I was like, "What does Khan Academy use for their draw thing?" And I tried it and I was bad at it. That guy, I don't know how it took too much learning, but I literally was like, "I love that black background with all the colors. I wanna emulate it." Uh, but so 5', 3', then the other one, opposite. 3' hydroxyl to 5' phosphate. They're anti-parallel to each other. They have directionality. They have handedness. That's important, the fact that they run anti-parallel to each other. Um, so here I name them. We have the 5' to 3' and the 3' to 5. It's riveting. Absolutely the best name ever. And then this, I've circled the bond that we are trying to make when we're making RNA. When RNA polymerase is adding the nucleotides, this is the bond that is being formed between them. And how we do that is pretty cool. So this is our template DNA, and then we have our RNA nucleotide that we have already incorporated into our mRNA, as denoted by this beautiful orange squiggly. This right here is this is an NTP, a nucleotide triphosphate. So this is gonna be our new nucleotide that's gonna be added into our mRNA. Um, and you guys are already super familiar with a, modified RNA nucleotide called ATP. This could be ATP right here. ATP is used for energy currency, obviously, but it is also incorporated into, uh, RNA. So if this is our new base right here, this is what's gonna be incorporated. It's the nucleotide with 3 phosphates. So how do we do that? What's first gonna happen is these, electrons right here on the oxygen, they're gonna perform what's called a nucleophilic attack on the phosphate. Um, this oxygen is super cool with giving up these electrons. It is, electronegative. And this phosphate is actually slightly positive because it's surrounded by oxygens that are stealing the negative charge. So that's how that's able to happen, this nucleophilic attack. So once that happens, some electrons on the phosphate are going to be kicked around, and that's going to result in 2 of the phosphates being literally just kicked off. So as these electrons attack the phosphate, the electrons in this bond are going to go with this oxygen, and it's going to kick it off completely into these two phosphates right here, the pyrophosphate. And in addition, our new bond is going to form between this 3' hydroxyl and the 5' phosphate. So that's how that's made. And you might be able to see already why only one of the DNA strands can be used for the template. That might be clear to you right now, but of course we're going to look at it anyway. I'm going to try to use the 5' to 3' as my template. All right, here we go. So this is my template, 5 to 3, which means that the growing mRNA, it's anti-parallel, so it's 3 to 5. I'm going to attach my new nucleotide, except no, I'm not, because plot twist, you can't. You just can't, you can't do it. It won't happen. Basically, all of these are already happy. They're happy with their lives. They are not going to react with each other, these two phosphates. Even these hydroxyls right here, which are, you'd be like, well, why can't the hydroxyl just flip around? And then, no, because there's so much potential energy stored in these three phosphates right here that's what makes these hydroxyls unreactive. So basically, in the simplest terms, all of these chemical components don't want to do anything when in the presence of each other. So we super can't use the 5' to 3' DNA strand as our template because this no work. But as a consolation prize, we'll call it the coding strand because the sequence of the coding strand is actually going to look pretty identical to the mRNA, as you'll see later. So it's a little consolation prize because we can't use it as the template. We have to use the 3' to 5' because right here, 3 to 5. The new base that comes in, they're anti-parallel. So it comes in oriented correctly with its phosphate across from the hydroxyl, and we can just do the frickin' thing we just did and make the new mRNA like this. So that's why it's the same strand every single time. We always read the DNA 3' to 5', and then we add the RNA nucleotides 5' to 3'. I hope that's not too confusing, the reading versus the adding, but this is why we absolutely have to use this strand as our template. So it's our template strand. All right. Let's move on with our lives. Now that we know the chemistry and now that we know which template, which DNA strand is going to be our template, we're going to frickin' move on and we're going to add the nucleotides. We're going to elongate. So to kick that off, TF2H is going to phosphorylate the tail or the C-terminal domain of the RNA polymerase II. Now this is something I've mentioned this before. There are some things that I think I've probably mentioned in every lecture, and one of them is definitely phosphorylating and dephosphorylating. It's just when you do that to proteins, it changes them. It changes their abilities, whether that be inactivating or activating them. In this case, the addition of these phosphates is going to make it so that the polymerase can disengage from all of these proteins at the promoter so that it can get on with its life and add nucleotides to its heart's content. It is also gonna the phosphates are also gonna allow other proteins to associate with it that are gonna prevent it from dissociating while it transcribes. And they're gonna do some other special things in a little bit. But basically the big thing right now is that when the tail is phosphorylated, it's moving on with its life. All right, so literal transcription. This is our coding strand. HEGTCCACT. We know our DNA alphabets, so we know what the template strand is going to be. Amy, take it away.
Oh. B, A. Yay.
Very good. Our
I
DNA
did.
alphabets. Good job.
I was like, wait, is the mRNA
We're going to do that too. But good job. Amy was totally 100% right. So that's our template strand is TACAGGTGA. Now we're going to use that template to make our new mRNA. Take it away.
Okay. Trying to remember.
You got this.
It, it, U T C U.
Really close. You swapped it.
Okay, so
It
A goes.
Yeah, A goes to
Okay,
yours still,
okay,
yeah.
sorry.
It's okay.
I didn't know which one of them.
Yeah. That's okay. Good job. Um, so that's our new mRNA. Now you can see why the coding strand is called the coding strand. It literally is the exact same sequence except it has T instead of U. You did great. You did great. So yeah, that's going to be the sequence of our example mRNA. Okay. So now that we have our we're in the elongation, we're adding the nucleotides. This is what our new mRNA is going to look like. That's not all. There's we can't just transcribe the mRNA, or we can't just transcribe. We have to do some things to the mRNA before it's actually considered mRNA. Because right now it's just pre-mRNA. There's a couple other things, a couple other changes that we need to make before it can just go off and party in the cytoplasm. And those things are encompassed by RNA processing. And this occurs basically during elongation. So what we need to do is we gotta cap it, splice it, and signal it. These are the things we gotta do before it can leave. So the first thing that's gonna happen is we're gonna cap it. As in, we're gonna put a hat on it. Remember those phosphate groups that we put on the tail of the polymerase? That is also gonna allow the binding of this picture shows just one. It just shows the one capping protein for simplicity's sake, but it's 3 enzymes that are gonna bind and add a 7-methylguanosine to the 5' end of our mRNA. This is very important because this 5' cap says this is an mRNA. This is not any other type of RNA, because there's a lot of other types of RNAs. But this signifies that I am an mRNA and I need to be translated into a protein. The 5' cap is also really important for later, when we do translation. It's so that the ribosome knows where to go, basically. So that's why we need to put a little hat on the mRNA. The next thing we need to do is splice it. We have a lot of junk DNA in our genome, and we don't need that, basically. They're called introns. It doesn't code for anything. And it's called junk DNA, but apparently there's some emerging evidence that, like, maybe it does something, but we could just call it junk. And we want it out of there. The exons actually code for proteins, and we want to take the introns out and splice the exons together. Because both, for whatever reason, we transcribe it all. We transcribe the introns and the exons, and then we take them out. I don't know why we do that, but it all gets transcribed for whatever reason. And this process is really freaking cool. It's done by the spliceosome, because of course that's what it's called. And it's big and chunky, and there's a lot of different proteins involved. This is the guy that's gonna it's not even just proteins, actually. It's a bunch of proteins and RNAs. So sometimes RNAs chop up their siblings. So that's what we're gonna do here. So the first thing this is so cool. A part of the spliceosome is going to bind to a section called the branch point. That branch point is pretty much always an adenine nucleotide, and then it is succeeded by a bunch of pyrimidines, which are U or C. Those are pyrimidines. So there's UC, UC, CC, UU, UCC, like a bunch of those, and then randomly an A. That's how the spliceosome knows, all right, this is the branch point. Another protein from the spliceosome is gonna bind to the 5' splice site. So there's something also in the sequence that lets the spliceosome know that, yo, this is the 5' end of the intron, and I need to bind there. So we're prepping for the splice. Then more parts of the spliceosome will bind, and they'll bring the two parts closer together. The branch site and the 5' splice site are gonna be brought together, and they're gonna catalyze the connection between this branch point and the 5' splice site. And the result is gonna be something called a lariat. I think that means lasso, and it looks like a lasso. So once this bond has been formed, now there's this really sexy looking OH on the 3' end of this exon that's just exposed now. And so the spliceosome is also gonna catalyze the connection of this with, and it doesn't show it here, but there's a phosphate here, a 5' phosphate here. It's gonna catalyze the connection between those two things. And as a result, this guy is bye-bye. And it gets degraded, and its nucleotides will be used to make something else. And then the exons are now together. So really cool. Really amazing how this happens. Now interestingly, we have about 20,000 different genes, but we can make way more proteins than that. I don't think we have a number yet, but we can definitely make like more than 100,000 proteins. And that's due to something called alternative splicing. So let's say we have 3 exons here, exon 1, exon 2, and then the red is exon 3. Sometimes things get spliced differently. So let's say in this case, the spliceosome instead splices out exon 2, and then exon 1 and 3 get spliced together. That's how we can make way more proteins than actual genes in our genome. I think the best example, there's a gene called, it's TTN, and it codes for titin, Titin. It is the biggest protein, the biggest gene, and it holds the record for the biggest exon. It's a big guy. And it codes, I think it's like a type of like muscle protein. There are so many different splice variants for that guy. So that one gene is able to code for a bunch of different proteins just because it's mixing and matching these exons, basically. This is not always a good thing. I'm gonna talk about something later where incorrect splicing basically can cause some problems. But alright, the last thing before we can send our baby to the cytoplasm is we just add a bunch of adenine nucleotides to the end of it. An enzyme called poly-A polymerase adds like 200 A's. 200 A's to the 3' end of our mRNA transcript. And this is its ticket out of the nucleus. In order to enter and exit our nucleus, you need to have fucking tickets. You need to have, like, your papers. You need to make sure that you're allowed to enter and leave. So this ensures that the mRNA is actually allowed to to leave the nucleus. And then when it's outside the nucleus, this tail of a bunch of A's also prevents it from getting, like, degraded immediately. Because the cytoplasm is really harsh. There's lots of enzymes there that fuck shit up. So it's good that it has this tail so that it doesn't die immediately, basically. So that's RNA processing, and it takes place during elongation. It's very efficient, so that by the time we are all processed, we're mature and we're ready to go. We're gonna take a little cool down. I'm gonna show you a video in a second that's really cool, but first, transcription factors are really important. It's really important that they do their job correctly. And it isn't just like, I know I talked a little bit about epigenetics, but it's not just like these really it's not just odd jobs, basically. Transcription factors do everything. And the example I have here is thyroid hormone. So thyroid hormone, it's involved in like metabolism and stuff like that. So when the hormone binds its receptor in your cell, it attaches to the receptor and both of them go straight to the nucleus and they literally bind directly to your DNA. And when they do that, they recruit a bunch of co-activators, so things that are going to open the chromatin and increase transcription. And as a result, more proteins are going to be made, and those proteins are probably going to be involved in lipid metabolism. So this is just something that happens all the time, and it's normal. And this is a very classic example. I think this is also how the sex hormones work. They have nuclear receptors. This is this opening and closing of chromatin. This is just it's just it's so cool and so it's happening all the time. And these trans factors also affect they do everything, but like they are needed for correct cell division, development, immune responses, and metabolism. I mean, they fucking do everything. Um, so real quick, we're gonna take a look at something that's gonna help kind of visualize everything we just looked at. So this is a fan-fucking-tastic animation. Do I have it on 2x speed? Yeah. So this is DNA being packed into chromatin. These are nucleosomes that are just being continually condensed and closed off. And eventually, and they coil on themselves, coil on themselves, coil on themselves. That's how they become chromosomes. So this is just a bunch of that, a shit ton of that, and it's so cool. And yeah, this is a real also before it was an animation, but this is I think an actual cell dividing. That's replication. Don't care about that. I just want to go to. Okay, so this is an example of like so vitamin D, the little guy comes in, it activates this protein so that it turns on gene transcription. I mean, it's that simple. This is kind of like what thyroid hormone was doing, because it didn't show it there, but the vitamin D was also actually connected to a nuclear receptor, but it's that easy. This is transcription happening in RNA polymerase. Wait, you guys might hear the sound on this. Oh, well, let me skip this. So this is a nucleosome. You guys, wait, here's the sound. This is a nucleosome, and those are the different components of a histone. Yeah, the sounds are just weird. This DNA is, as you can see, it's kind of farther apart, so it's actually actively transcribing. It's possible to actively transcribe it. This is another way of saying I use the term histone modification. These are the chaperones that just fucking delete, which allows now for transcription to take place because they took the nucleosomes out. This is, I think that's also a chaperone. Yeah, just moving it. And then yeah, this is yeah, so there's all these signals that let proteins know like this needs to be condensed or don't transcribe this or whatever. And it's different in different cells, you know? That's how we distinguish them. So yeah, just nice cool down, nice visualization by thank you to these people for doing that. A little body horror, yes, a little bit. All right, so that was a nice visualization of what we just did. So now we're going to go into the next step, which is translation. And we call it translation because, I mean, DNA and RNA are pretty much the same language. They're all nucleotides. But now we're going to take our nucleotides, our nucleic acid code, and we're going to turn it into the protein code, which is amino acids. Lost in translation. Oh my god. Mutations. Mutations, that's how we get lost in translation. But yeah, so transcription, we're transcribing the same language. Translation, we're nucleotide to amino acid. Completely different. So let's frickin' do it. This is our hamburger. This is our body hamburger. This is a ribosome. Um, Ribosomes and tRNAs are gonna be our main characters in translation. Um, so, um this is what a ribosome looks like. As you can see, there's 3 sites. E is for exit site, P is for peptide site, A is for active site. Um, so we have entrance, growing peptide, and exit, as we will see. tRNAs are they're RNAs, so they're single-stranded and they're shaped like a clover right here. And each tRNA is going to be connected to an amino acid. They are going to bring amino acids to the ribosome and add to the growing protein chain. And on the bottom here is something called an anticodon. The anticodon is a string of 3 nucleotides that corresponds with a codon, which is 3 nucleotides on the mRNA. So the codon's on the mRNA, anticodon is on the tRNA. Um, so and since each tRNA is associated with an amino acid, that means that each of these 3 these groups of 3 nucleotides directly correspond to an amino acid. And then peptide bonds, this is what's being catalyzed by the ribosome. The peptide bonds are the connections between the amino acids that make the full protein. So let us translate. More Khan Academy. Again, I'm so great at drawing. So this is our small ribosomal subunit, which is the bottom part. And it's got the E, P, and A site. We also, there are more proteins bound to the pre-initiation complex, but these are the most important. We have EIF1A, which is blocking the A site. And then we have EIF1, which is eventually going to help the mRNA be threaded through the small ribosomal subunit. And then both of these guys together are blocking the large subunit from binding because we're not ready for it. We're not ready for the large subunit yet. We still need to include the star of our show, the initiator tRNA. tRNAi. The initiator tRNA is always, always, always bound to the amino acid methionine, and it is always, always, always the first amino acid in a protein. Always. And that's because, so right here, the start anticodon is UAC, which means our start codon is AUG. AUG is our start codon. So it's always the first one, and what makes it so effing special is, 1, it is the only tRNA that binds directly to the P site. Every single other tRNA has to go in through the A site first. But this guy's special, gets to go in the P site first. Another special thing about the initiator tRNA is it's the only one that can bind EIF2. EIF2 brings the tRNAi to the P site. And it's also bound to a GTP. When everything is here, when like all the proteins and when the initiator tRNA are all here, that is what's gonna allow the mRNA to finally be threaded into the small ribosomal subunit. And then it's hamburger time, baby. As soon as this base pairing happens, as soon as the start anticodon binds the codon, that is gonna cause a change in affinity. That is gonna cause GTP to be hydrolyzed. And we're gonna see this a couple times, so I'll just say right now. GTP hydrolysis, it's a chemical reaction that causes shape changes, affinity changes, things like that. So in this case, it causes the affinity of this guy to be lowered, so it leaves the tRNA, and so does EIF1. Boom. Eventually, the EIF1A is going to be kicked off when the large ribosomal subunit comes in. So, boom. We are assembled. We are freaking ready to do translation. Our tRNA is in the P site, our A site's open, we got our mRNA. Let's go. So this is now we have a couple polypeptides, right? So this is no longer the initiator tRNA. It's a completely different one that is now responsible for the growing polypeptide chain while in the P site. So we can assume that this brown guy is the methionine because it's always the first one. We also have a new tRNA in the A site. So as soon as the binding here happens, oh wait, no, it's brought here by someone. EF1A. We love these names. So good. So this guy brought this blue tRNA to the ribosome. And as soon as the base pairing happens here, this is also going to have the large ribosomal subunit is going to hydrolyze that GTP. And the EEF1A, that's going to cause it to rotate off of the tRNA. And as a result, there's going to be this compensatory rotation by the tRNA. It's going to look like this. So now it's slanted. As a result of the EEF1A going like that, this is now going to rotate in the opposite direction because, physics and shit. So when it's slanted like this, now its amino acid is closer to the P site. And that, now that they're closer, that makes it way easier. Now the large ribosomal subunit is going to catalyze the formation of a new peptide bond between these two guys. And it's going to be given to this tRNA that's in the A site. Now, this is, it's not depicted in this picture, but these two guys are slanted right now. And when they're like that, that's called the hybrid state. And there's a protein that really, really likes ribosomes and tRNAs that are in the hybrid state. And that's EEF. Whoops. Oh, it's in the... Okay. I skipped the ratchet. How could I skip the ratchet? Okay. So, the slanting, before that other protein binds, the slanting is actually going to cause the large subunit to ratchet over. So now, this is the hybrid state. The P site of the large subunit is over the A site of the small subunit. So that's why we're saying this tRNA is in a hybrid state. It's in basically two different sites at once, which means this guy's in the E site and the P site at once. Now that we're super definitely in the hybrid state, that's when a protein called EEF2 is going to bind. And this, I wish I could find an animation of this because what this protein does is really, really cool and mechanical for anyone out there who likes fucking gears and pawls and shit. It acts as a paw. And it prevents the ribosome from moving backwards. And when its GTP gets hydrolyzed, it causes the small subunit to ratchet over under the large subunit. Really machines, right? Like, this is so cool. Like, really fascinating shit. So now that both of the subunits are back in sync with each other, now this guy is fully in the P site. This guy is in the E site and so gets ejected. And now the A site is ready to accommodate a new tRNA. And the cycle keeps repeating until the protein is complete. So please take note of all the signaling here. Like it's just these molecular signals. like the attachment of the initiator tRNA, the binding of it to the mRNA is what sorry. Yes, the binding of the initiator RNA, the very first one, to the mRNA, that's what signals the large ribosomal subunit to come in. And then the hybrid state, the fact that the tRNAs are in a hybrid state is what signals the EEF2 to come in and like ratchet the ribosome so that it keeps moving. Um, I mean, it's just so fascinating to me. All of these protein RNA interactions that are all um moving these processes along just by existing. It's so goddamn cool. So this keeps going until until the ribosome reaches a stop codon. There are specific 3 nucleotide codons that's just they don't code for an amino acid, they just say, "Boom, we're done. Wrap it up." Keeps going until it reaches one of those. Um, so let's learn that language though, the language that we just translated. So the start codon with the anticodon, um, there there are 20 amino acids, and as you can see, there's more nucleotide combinations. And I won't expound on this, but that's because of something called wobble. Basically, the third, if my hand's a tRNA and this is AUG, this third nucleotide is whatever. It doesn't give a shit. So like that's why like serine and proline have a lot. As you can see, as long as there's a UC, the tRNA doesn't give a shit. So that's why we have all these combinations. It's fucking crazy. Um, so let's start with the one that we already know. We know AUG. It's already in red for us. Boom, it's methionine. So the next one is UCC, that's gonna be serine. And then ACU is threonine. Easy. This is how the codons get read. This is how the amino acids continually get added to the growing polypeptide chain. And there are there's lots of fail safes for both transcription and translation. Like there's proofreading, and all that. And that's because it's really, really important that everything here gets made correctly. We don't want to make mistakes. Because amino acids can be very different from each other. They can differ in pH, size, charge, polarity, hydrophobicity. And so they contribute because of that, they contribute a lot to the final shape of the protein. They affect how the protein folds based on these amino acid interactions. And if it contributes to its form, it contributes to its function, as we know. So even one wrong amino acid can mess shit up. Even one wrong nucleotide can mess shit up. And the best example of this is sickle cell disease. So, we have the change of just literally one nucleotide in the beta globin gene. So here's the normal one. We have a GAG. Is GAG? Wait. Yeah, okay. So this is the normal one. We have a GAG, which codes for glutamic acid. But then things get changed to a GTG, which becomes GUG, and you have a valine. This is very bad. Because these amino acids are completely different from each other. I mean, glutamic acid is polar and hydrophilic, which means that it would much rather react with things other than itself, like water, which is good, especially for the function that we're trying to do with, carrying oxygen in blood. Valine, on the other hand, is nonpolar and hydrophobic, which essentially means that it likes to interact with itself too much. So it will polymerize with itself and cause all of this rigidity and eventually pain. So On the left, this is obviously a nice normal erythrocyte. It can carry oxygen normally, it doesn't aggregate. Whereas here we have the classic sickle cell shape. So they're very rigid and they don't carry oxygen efficiently. And that's literally just because of this one change to a completely different amino acid. It's really it's terrible. Like it's just crazy to me. Just making a protein right is really important. Um like there's so much that can go wrong. There's so much that can go wrong if the transcription factors are incorrect or if the chromatin remodeling enzymes are incorrect. Like those types of issues can cause developmental disabilities. Parkinson's and Alzheimer's are all like incorrectly made proteins that all aggregate and form plaques. Uh Even like pathogens can also disrupt these processes, like diphtheria, for example. Um Diphtheria inactivates EEF2. So it just blocks translation. It stops proteins from being able to be made. It's crazy how oh my god, everything can go wrong with just like one little thing. It makes me sick, but I love it. Um we're gonna make our own protein. It's gonna be so sick. We're gonna go through the UCSC genome browser. But first I do wanna again, I want you guys to just see this I think this video will help you visualize translation. Um, so this is the mRNA. It's coming out of the nucleus into the cytoplasm where it's gonna shack up with a ribosome. All these green things flying around, those are all tRNAs. And then this is the growing polypeptide that's coming out of the top of the ribosome. Um, so here it's shacking up, small, large subunit. Um, the little tRNAs, the red, that's the amino acid that they're carrying. And then this A site, P site, E site. It enters, it adds to the polypeptide chain, and then it gets kicked out. Wow, it's showing us a lot of angles here. Um, it's really cool, and you can kind of see here. I think it'll show in a little bit. Yeah, the polypeptide it's starting to fold while it's still being made. So the amino acid interactions are, like already happening. They're already taking place as the protein is still being, as peptides as amino acids are still being added to the polypeptide. So yeah, this is translation. And you can see, yeah, the little guy coming out the top. Love this shit. All right, guys, let's make our own protein. I'm gonna show you guys some really cool shit right now. Um, all right, we're gonna fucking do it. I'm so excited. All right, so uh, this is the UCSC genome browser. It's the genome browser. We're gonna look at a protein of my choice. I have chosen phenylalanine hydroxylase, PAH. It's encoded by the PAH gene. Please don't be down. Please don't be down. Yes. Okay. Phenylalanine hydroxylase is really important. It breaks down the amino acid phenylalanine. It breaks it down into tyrosine. That's very important. Because tyrosine is how you make dopamine. It's how you make adrenaline. It's how you make thyroid hormone. You need it. Also, phenylalanine in high amounts is brain toxic. So you want to consistently be breaking it down. You really want this protein to be made correctly. So what are we looking at here? This is chromosome 12 right here. This whole guy right here is chromosome 12. Um, and that's where the PAH gene is located. And it is specifically on this red mark. Boom. This is just exactly where it is on the chromosome. 12q23.2, that just means it's on the Q arm, the long arm of the chromosome. Region 2, band 3, subband 2. Lots of bands and subbands and subregions, but this is where it is. um let's we're gonna look at its code. This is how many base pairs it is. 80,356 base pairs. Just for us. So let's look at I'm gonna open this in a new the whole genomic sequence. I don't want the promoter. This is fine. So the exons are gonna be uppercase. All the things that are actually gonna code for the protein are gonna be uppercase. So let's take a look. Wow. Okay. So there's exon 1. Where is it? It might not have a lot of exons. I think it has 11 though. There's another one, the uppercase. 2, I might be missing it. 3, I saw 3, 4, 5, 6. Wow. So this is the about 80,000 freaking base pairs. And look at how much of it is not all the lowercase is introns. Look at how much of it is not even going into our shit. That's so much. All right, let's look at the mRNA itself. Please don't be down. The genome browser is down a lot, I feel. Okay, cool. So this is our mRNA. So what you might notice is it starts with ACT and not they put the T's in here for no, so this is the coding strand basically. I don't know why they do that, why they put the coding strand instead of the actual mRNA. But this is not methionine. It should be this should be ATG. So that's because they for whatever reason they put the 5' untranslated region here, I think. But that's okay. We can find the start codon because I have it here. I just have to know that. Here we go. ATG TCC. And we'll find the start of it. It's actually not that far into the mRNA. But this is everything that's gonna be translated into a protein. The protein itself is 452 amino acids. And as expected, we have methionine that starts it. And plot twist, the other two nucleotide, the other codons that I use as the example with the serine and the threonine were from this gene. Um so let's check the third one. Let's or the fourth one, alanine. Is alanine should be GCG. I think it is. Yeah, okay, cool. So this is right. I didn't think it was wrong, but I just wanted to show. Um so this is our mRNA transcript. This is our whole ass 80,000 nucleotide frickin' gene basically. Now this is another interesting thing. It says strand minus. That means that like and you can tell because this is ATG, they're showing us the coding strand rather than the template strand. SNpedia, 23andMe, some companies show the minus strand. I think it's because it's the coding strand. So they want to show you an idea of what the mRNA looks like. But it's interesting that it's not the plus strand or the template strand. So yeah, these are visualizations of the DNA, the mRNA, and then this is just the protein readout. So let's look at the protein. PAH. Yep. Look at this guy. Look at this beautiful. Wow, look at all the chains. These different colors are representing the different chains. So this is a complex. The gene codes for the different like chains of the protein. We can also look at confidence, which is how correct do they think that this is? This is pretty good. The dark blue is like, yes, we definitely know that this is where it is. This is the catalytic domain too, so it's good that we know that. I like looking at the chain. So earlier I was talking about when we make proteins wrong. If you make this protein wrong, you probably have a disease called phenylketonuria or PKU. If you've ever read the nutrition facts on diet soda, you might have heard of PKU before. But that means that you can't break down phenylalanine, which is really, really bad. I mean, it's severe intellectual and developmental disabilities. Um, and there is treatment. You have to avoid phenylalanine completely. You just cannot eat anything that has it. That's why diet sodas have the warning, I guess. And you have to eat a lot of tyrosine because you have no way of really making that yourself if you don't have PAH. Um, and I just wanted to talk about 2 mutations uh in this guy that I thought were interesting. So one of them is 408. Here we go. Whoa. So you see, it might be a little small, but it says 408. Yeah, 408 R to W. This is the most common mutation in PAH, and what this means is R is arginine. The amino acids also have like a single letter, as you saw. R is arginine and W is tryptophan. So what this is saying is in the 408th position of this polypeptide, the mutation changes from arginine to tryptophan. Now, the orange is highlighting where that is. And remember, these are different subunits. So the protein basically binds together to form a tetramer. So there's 4 of them. This is in the catalytic domain of the protein. So if you have the mutation, it doesn't do anything basically. So even if you are making the protein, it's not going to do anything because of this mutation. Um, and that has to do again with how different these amino acids are. This will tell us right here. Arginine to tryptophan changed from a large size and basic to large size and aromatic. So that's very different. Tryptophan is very like has an aromatic ring, and the chemistry that arises from changing from arginine to tryptophan just makes it so that it's completely useless. It doesn't do anything. And that is that's very bad. Um, so yeah, if anyone wants to see any other genes for no reason or any other protein structures, I'm certainly happy to oblige. But for now, we're gonna go. I want to talk about one more mutation in uh in this gene. Oh, this was just in case the browser was down. um, so this is the one I just talked about. It's in the catalytic domain. If the protein is made wrong, or it's like because of the wrong amino acid or missing amino acids, uh, then the location, like it's in the catalytic domain, that's gonna make it can't do the literal reaction that it was made for. It can't break down phenylalanine. Uh, and it can also cause it to misfold, and we know that when proteins misfold, they aggregate and form plaques, and that's bad. So this is just mutation basics, change amino acid, change nucleotide. In sickle cell, it was both of those things. This can lead to changes in protein shape. Um, there can also be stop codons where there's not supposed to be, so you can end transcription prematurely, and then you make a protein that's way too small and doesn't do anything. You can call, oh my god, a shift in the reading frame. These tend to be really bad. So like if you have um like let's say you have AUGTCCTCC or something, uh a frame shift maybe because of a deletion, it would be AUGCCTCC. So now completely different codons, completely different amino acids being made. And then lastly, this is the one I'm going to talk about. Incorrect splicing. This is fascinating. So this is the C165 deletion mutation of PAH. So in the DNA, the 165th nucleotide is deleted. Normally, And I said this before, remember, this, the branch point when you're splicing is always an adenine. And it's supposed to attach to the G of, the beginning of this intron. And like I said before, I said that there's things that signal, like, the splice sites of the intron. Usually it is a GU, something like this. So when it's normal, it splices out intron 12 so that you have exons 11, 12, 13. But if you delete this nucleotide, it causes a frame shift. So now there's an A here instead of a G. So this A, this branch point, it doesn't recognize this. It doesn't know what the fuck this is. It's gonna go to the one over here. It's gonna just skip this one completely. So now instead of having 11, 12, 13, you just have 11 and 13. That's wrong. Uh, this single nucleotide change caused the splicing to be incorrect, and now the final protein it's 52 amino acids short. Again, this is one nucleotide. Things go wrong, and it's crazy to me. It's just crazy to me. It's fine. I'm fine. I think that's it. Yeah. Um, this was great. Like I said, shout out Molecular Biology of the Cell 6th edition. This is my shit. I'm just showing my dedication to my craft. Like hanging out with Rosalind Franklin. And just, and you see, I've been asking this question a long time. How does life, I don't understand. I mean, I understand, but I also hate it, like, a lot. I hate it and I love it, and I'm just so dedicated. I even used to do my OChem homework with my goggles on because it would give me powers. And that's just how it works. So, alright. This was easier than I thought. Like, this is my shit, but some of this stuff, it's, like, it's so fucked up and it's so complicated that it's, like, I feel like I'm wearing a tinfoil hat while I'm trying to explain it to people. But everything's fine. I think I also, like, I cut a lot out, so I'm definitely gonna do a follow-up lecture on genetics because I think this is something that people are really interested in. I really wanna talk about genes and alleles and, like, gene expression and things like that. Um, do fun things like, "How do you cross your hands?" and, "Can you do the tongue thing?" and, "Do you have a widow's peak?" Like, I love that shit. Um, I I like talking about the individual genes that might affect those things. And, you know, even in this lecture, I really wanted to talk about the difference between, like, molecular and Mendelian genes, but I could do that another time. So, yes. Just in time because 'cause I'm starving. So, I appreciate, um appreciate all y'all. This was great. Definitely the highlight for me was, looking at protein and going to the genome browser. I love genome browser. Thanks, David Hostler.